My name is Hannah Stesney, and this is my first presentation. My article title is Functional Connectivity Between the Temporal Parietal Cortex and Cerebellum in Autism Spectrum Disorder. This article was published in 2017 into a journal titled Cerebral Cortex. Previous research has determined common deficiencies in the cerebellum for individuals having autism spectrum disorder. The cerebellum is the part of the brain best known for its role in motor coordination. It is also one of the key brain regions affected by autism spectrum disorder that we know of as of now. It is also the most highly studied area of the brain for these individuals. In the context of autism spectrum disorder, the temporoparietal junction is a far less studied area of the brain. However, previous research has also determined that adults with high-functioning autism spectrum disorder have altered white matter integrity of the temporal-parietal junction. It is important to note that both the cerebellum and the temporal-parietal junction are involved in many social and cognitive functions. Looking at this image, we can see that the cerebellum is located in the bottom right, purple region. However, the temporal-parietal junction is going to be where the parietal and temporal lobes meet. With all that said, why do this study? It is important to do studies such as this one because there is an increased prevalence of autism today. That could be due to increased diagnosis, research, and awareness. However, the fact is that more individuals are now being diagnosed with autism spectrum disorder than ever before. Now is an important time to research and further understand this disorder. From that, better diagnosis and therapy strategies can be developed, thus providing these individuals with an increased quality of life and care. Now, what is the impact of this specific research article? Currently, little is known about the role of TPJ in autism spectrum disorders, diagnosis, or therapy. From this specific research, new strategies for non-invasive brain stimulation therapies can be developed, and an overall increased understanding about brain structures correlated to an autism spectrum disorder diagnosis. The dependent variable was the differences in brain structures, and the independent variable was whether or not the subjects had autism spectrum disorder. And the researchers hypothesized that there are abnormalities relating to the TPJ and cerebellum specific to autism. This study used resting state fMRI data, resting meaning that the subject wasn't given anything that would stimulate specific parts of their brain, and an fMRI is a way to measure blood flow, specifically how blood flow changes with brain activity. All of their data was collected from the Autism Brain Imaging Data Exchange, which is a database providing resting state fMRI data inclusive for individuals within the entire spectrum. The researchers controlled which data was used and only accepted data derived from the same scanners. This allowed researchers to minimize variability from different fMRI scanners. And to the right is a picture of what these scanners may look like. The population consisted of 60 males aged 10.2 to 16.8 years old. Of those 60 males, 26 had autism diagnosis and 34 were neurotypical. The researchers ensured that age and verbal and performance IQ had little variability between groups. A region of interest is the area of the brain that researchers want to focus on. In the image on the right, you can see the researchers' area of interest and specifically where their research was focused. After establishing a region of interest, the researchers then tested for differences between these regions between neurotypical individuals and individuals with an autism diagnosis. They did this using dual regression and permutation testing. First, the researchers studied differences in the functional organization of the temporoparietal cortex between subjects with and without an autism spectrum disorder diagnosis. Then the researchers studied how the temporal-parietal junction was connected to cerebral zones, and last, any abnormalities in autistic subjects' connectivity between the temporal-parietal junction and cerebellum. What did they find? One of the main questions was whether the organization of the temporoparietal junction was altered in individuals with autism spectrum disorder. Interestingly, after comparing the data from participants with and without autism spectrum disorder, the researchers found no significant abnormalities in the organization of the temporoparietal junction. On the bottom of this slide is an image of what the organization of the TPJ should look like, with 5 regions on the left and 6 on the right. And as mentioned previously, this organization remained intact for individuals with an autism diagnosis. When researching connectivity between the TPJ and cerebellum, abnormalities were present. The researchers found one striking difference between the connectivity of these two brain regions for individuals with an autism diagnosis, that being that the right dorsal TPJ showed much less connectivity with the left crus2, which is a region of the cerebellum essential for social behaviors such as mentalizing, cognition, and multimodal integration. These findings highlight the importance of further research regarding the connectivity issues between the right dorsal and left crus2. Additionally, the researchers propose that this abnormality may contribute to social deficits associated with an autism diagnosis. This figure shows the connectivity abnormalities present within autism. The upper image shows the right hemisphere of the TPJ, and the bottom image shows the crus2. The locations highlighted in this figure were shown to have abnormal patterns of functional connectivity for individuals with autism. In conclusion, this research article highlighted a new connectivity abnormality within the brain structures of individuals with autism spectrum disorder. Importantly, regarding individuals with ASD, these findings were in a rarely researched area of the brain, the TPJ. Therefore, it is important that further research is conducted to better understand these results, the impacts, and future therapy or diagnosis measures.